Friends, Tanu is asking at what time class will be over today. Yeah, today it is only 8 o'clock right now. If you sincerely listen to me, I think the class will be done by 9 o'clock or 9:30, maximum 9:30. Okay, so another 1.5 hour class will be done and it will be done. Okay, so please listen here right now. Now, post transcriptional modification, we are already having an RNA here right now, guys. Just now RNA is formed here, and this RNA will be from 5' to 3'. We all know that RNA will be like single stranded RNA here right now, guys, 5' to 3', and this RNA which is just now formed here is known as hnRNA. Already I told you in the overview. तो चलिए से इसको modify करते हैं. Let us modify this one. Please write down there. Extremely important topic, and you extremely important topic. And my dear friends, you can always expect one question from here, sir. So hnRNA will be from 5' to 3', sir. Okay, one of the modification will be taking place at 5'. 5' is like head. On the head you'll be wearing cap, इसीलिए उसको कहते हैं 5' capping. That is the reason why it is known as 5' capping. And in 5' capping, the substance that will be attached here will be the 7-methyl guanosine. In 5' capping, what is the substance attached, sir? 7-methyl guanosine. 7-methyl guanosine, sir. ठीक है. So 5' capping means there is a substance attached, 7-methyl guanosine. हो गया, sir. One modification done. I told you now it is like just 2 minutes topic. And 3' ends like a tail. Tail पर यहाँ पर ना tail लगा दो. Attach one tail here right now, sir. So there will be a long tail of adenine, like many adenines attached there, sir. So because there are many adenine, many means poly. Adenine starts with A, that is poly A tail. Okay. So at the 5' end there will be 5' capping, and at the 3' end there will be a poly A tail. At the 5' end there will be 5' capping, and at the 3' end there will be a poly A tail. Poly A tail. Okay. And the last and final modification that you have to learn in the middle. In the middle, you'll be having some small parts here right now, sir, and those parts are actually known as introns. And then the part in the middle here will be the exons. So I write here I for intron and E for exons. And remember, introns will be like non-functional, and whereas exons will be like functional. So therefore, bolo bachcho kya hoga? Which one has to be removed off? Which one has to be removed off? Intron. So therefore, my dear friends, introns to be removed. Introns remove. And after removing the introns, ligate the exons, attach the exon. And that third modification is known as splicing. इसको हम कहते हैं splicing. Splicing means what? Splicing means removal of intron and ligation of exon. Removal of intron and ligation of exon. Splicing means what, guys? Removal of intron and the ligation of exon, and that is splicing. And important thing is it is done by an enzymes, and those enzymes are actually known as spliceosomes. And spliceosomes, they are also known as snRNPs. And snRNPs, if you read it like multiple number of times, it will be like snRPs. snRPs like that. Okay, now. So spliceosome or snRNP or SNRPs, these are the enzyme which is going to do what, sir? Splicing. Is it okay? हो गया. Modification number 1, 5' capping. Modification number 2, poly A tail. And modification number 3, splicing. After doing this, finally whatever RNA is formed here, that will be the fully modified mRNA. That will be the fully modified mRNA. If you are done, please let me know, sir. हो गया? Are you done? चलो sir. Now once you are done with this one, guys, the modification is done. And now, my dear friends, see, we'll do one thing here. Hello everyone here. Yeah, intron is not useful, it is non-functional. We'll remove the intron, sir. So now we are done with the transcription and we are done with the post-transcriptional modification here right now, my dear friends. And now, my dear friends, what we have to do is we need to study about translation. But you know, I'm seeing from last 5-6 years, guys, not at all a single question is coming from the translation process here. Okay, na? So I'll just tell you whatever are important topics from here onwards to be learned. Don't worry about translation, guys. Please don't waste your time in unnecessary things. I always advise. There is so much content available, so much subject available. Important thing is what to be learned, what to be left for the exam. Okay, na? So don't worry about translation. I'll teach you some important topics which are required here right now. So, okay, so please observe here right now, guys. Everyone look here. The first important topic that you have to learn is about the genetic code. Genetic code. First of all, what is another name of the genetic code? Genetic code is also known as codon. Genetic code is also known as codon. Codon. Now, what is the definition of the genetic code of the codon, my dear friends? Yes, right down there. It is a combination. It is a combination of 3 nucleotides which codes for an amino acid. It is the combination of like 3 nucleotides which codes for an amino acid. Now, what is the meaning of this one here right now? I'll give you an example, you'll understand everything. I think you have been learning about this one, guys. AUG. What is AUG, guys? Yes, adenine uracil guanine. Yes, it is a combination of 3 nucleotides. Okay, it is a combination of 3 nucleotides. Okay, it's a codon, and AUG will code for an amino acid, and that is methionine. Yesterday we have learned like 20 amino acid. Out of the 20 amino acid, methionine sulfur containing AUG will code for methionine. Are you getting my point here right now? Genetic code or codon will be coding for an amino acid. It is a combination of 3 nucleotides. Perfect, done. We understood the definition. And now, my dear friends, for example, if this is the RNA here, right? If this one is your mRNA, messenger RNA, okay, from 5' to 3'. Okay, remember on this we'll be having like many codons, and remember always the very first codon will be AUG. So therefore this AUG is also known as the initiation codon. Initiation codon, or else this is also known as the start codon. AUG is also known as initiation codon. And remember at the end of this always will be having the either UAA or UAG or UGA. Okay, na? So UAA or UAG or UGA, these 3 are together known as your the stop codons, or in some good English they are also known as the termination codon. And because they are not coding for any amino acid, they are also known as the nonsense codons, guys. Nonsense codon. So remember, in an RNA, mRNA, always the initiation codon will be AUG and the termination codon will be your UAA, UAG and UGA. Okay, are you done with that one here right now? And now, my dear friends, this is what they will ask you. इसलिए मैंने कहा ना आपको, okay, don't waste your time in unnecessary topics. Whatever is they're asking in the exam, just learn that, more than enough. ठीक है? And now this codon has got some special properties right now, guys. 4 properties. Okay, so let us study those 4 characteristic properties. Heading लिखो, characteristic properties. That's something important nowadays, guys. Remember, number 1, the characteristic property is a codon will be universal. Okay, what is the meaning of universal, guys? A same codon, okay, codes for the same amino acid in all species. Okay, a same codon codes for the same amino acid in all species. For example, I gave you the example as AUG here right now, guys. AUG codon will be coding for which amino acid? It will be coding for methionine amino acid. Now that AUG will code for methionine in human beings like you and me. AUG will code for methionine in animals. AUG will code for methionine in dogs. AUG will code for methionine in plants. In every organism, AUG, the same codon, codes for same amino acid in all species. Okay, that is the reason why a codon is universal. Number 2, Whenever you are writing some codon, for example AUG, UAA, and UGA, something like that, whatever codons you're writing, you are not allowed to use any punctuation marks in the middle. Some students are like, acha, sir said it is a combination of three, after AUG, comma, after UA full stop. No, you're not allowed to use the punctuation mark. So therefore, comma less, it will be comma less. And not only comma less, acha, sir asked not to use comma in the middle. Chalo, let us overlap that. You cannot even overlap. It will be non-overlapping. Non-overlapping. So therefore, you cannot use any punctuation marks in the middle, sir. No commas and all. And so therefore, non-overlapping. Okay. And number 3, remember the codon will be specific, guys. Specificity. And this specificity is also known as unambiguous. Now, what is the meaning of that? Yes, AUG will code for which amino acid? Methionine. So remember, it is specific for methionine only. That's all. It's not like yesterday AUG is coding for methionine. Today morning it is coding for glycine. By evening it will code for lysine. ऐसा नहीं होता है. Amino acid will not change. It is specific to particular amino acid. And number 4 property, a codon is degenerate. Degenerate. And how to understand this one with number? My dear friends, in total, how many codons do we have? We are having like 64 codons. How many codons do you have, guys? 64 codons. But in total, how many amino acids do you have? You have only 20 amino acids. Come on, guys. Ulta dekhiye. One amino acid can be, yes, one amino acid can be coded by more than one codon. There in total how many codons? 64 codons. So therefore less amino acids. So therefore I can say that one amino acid can be coded by more than one codon. Codons zyada hai. We are having like more codons. That is why that property is known as degenerate. One amino acid can be coded by more than one codon. That is known as degenerate. So my dear friends, nowadays trending questions you have to remember. Yes, a codon is universal, commaless, non-overlapping, specific, unambiguous, and degenerate. That is what they are asking in the exam nowadays. Is it okay? Are you all done? Come on. So this is one topic which is important for us, guys. Okay, codon wala topic. After this, which topic is important, guys? Please write on the heading there as RNA. RNA. You know we have got like different type of RNA. Messenger RNA, ribosomal RNA, transfer RNA, and all those different types of RNA. Out of that, just remember these points, sir. Can you please tell me, out of all those RNA, which one is the most abundant RNA? Which one is the most abundant RNA? Most abundant. It is your ribosomal RNA. Ribosomal RNA is the most abundant RNA. Okay, na? Because every cell will have ribosome, and ribosomes are like abundant ones. Okay, na? And which one is the smallest RNA? That was one more important question. Smallest RNA, that will be your tRNA, sir, transfer RNA. Okay, and out of all the RNA, my dear friends, don't remember like each and every RNA, just remember about the transfer RNA, sir. So please write down the heading as tRNA. Okay, let us learn about the structure of tRNA. Do you know what is the shape of tRNA? The shape of tRNA will be like clover leaf shaped. Shape of tRNA will be like clover leaf shaped. It will be clover leaf shaped. Clover leaf shaped. Okay. So therefore, please draw the diagram of the tRNA, guys. That's important. And in details to be remembered, this is 5' end and this will be 3' end. Okay. At 3' end, you have to remember the sequence. The sequence will be, yes, anyone, any idea? Come on. It will be cytosine, cytosine, adenine. Cytosine, cytosine, adenine. That is the question number 1. Chal. Question number 2. At the 3 dash end, it is going to attach amino acid. Amino acid will be attached here. So because it is accepting amino acid at 3 dash end, that arm is known as acceptor arm. इस arm को हम कहते हैं acceptor arm. At 3 dash end, it is going to accept the amino acid, therefore acceptor arm. Exactly opposite arm is for reading the codon. Therefore, this arm is known as the anticodon arm. Anticodon arm. So 3 dash end will be acceptor arm and the opposite arm will be the anticodon arm. And here, this part here right now, one more arm here right now, actually that is not needed. To be very frank, for your MCA exam, if you learn this acceptor arm and if you learn anticodon arm, more than enough, sir. More than enough. Now, for some students who are like very curious, yes, this arm here right now, will be dihydrouridine arm. Just remember DHU arm, and this will be like T. You remember symbol psi? T psi C arm. And if you are still unsatisfied, not satisfied, sir, I want more. Yes, sometimes present, sometimes absent. There will be one more arm, and that will be variable arm. कभी कभी होता है, कभी कभी नहीं होता है. Enjoy. But remember, my dear friends, if you are targeting your MCA exam, remember 3 dash and acceptor arm, and exactly opposite of that anticodon arm. So that is more than enough, sir. Okay, that's all. So in RNA, if you remember this much information, more than enough, sir. So it's like, you know, almost we are at the end right now. We are having like small topics. So codon completed, RNA completed. Shall we go ahead right now? Are you ready? Are you understanding, guys? Tell me yes or no. Anticodon arm means that is going to go and read the codon. वो जाकर codon को read करेगा. हो गया बच्चों? चलो यहाँ देखो सर. Now once you are done with this one here right now, my dear friends, चलो सर इसको खत्म करते हैं. This topic will finish it off. I told you very clearly. I told you learn transcription in detail, learn post transcription modification in detail. I told you very aram se slowly. Don't worry about translation right now, guys. Okay. From last 5-6 years, there is no question at all. They're not asking any question from there. Don't worry about that. Just now worry about this topic, that is the post translational modifications. ये पढ़ते हैं और इस topic को खत्म करते हैं. Let us finish this topic. So all like small topics. So please write down the heading there as the post translational modification. What is your doubt, Bharat? Bharat, I told you very clearly, CCA, cytosine adenine, is at 3' end. And whatever initiation you are telling me, initiation will begin from AUG only. And AUG will be at 5' end, not 3'. And second point, that will be mRNA, messenger RNA, not tRNA. Concentrate. Okay, welcome here right now, sir. Now welcome to the post translational modification. Subhash, अरे यार तुम लोग सुनते क्यों नहीं हो? I told you very clearly, don't waste your time in learning translation right now. They are not asking any questions in that. Don't learn the topic. Don't study the topic. Now, sir, for the last and final time, DNA is getting converted. Yes, DNA is forming your hnRNA, heteronuclear RNA. Heteronuclear RNA is modified towards a mRNA. Now this mRNA will come out of the nucleus into cytoplasm. mRNA is forming your protein. Okay, and now I am speaking about this protein modification, post-translational modification, important topic. You imagine one of the protein, that is collagen protein in our body, and you have to learn the modification of only the collagen protein. So, see, now here also some students will be like questioning. Actually, na panga ye hai कि in our body everything is like almost protein only. Just enzymes nature, proteins. What is the nature of hormones? Protein. Transporter? Protein. Receptor? Protein. And ion channel? Protein. Muscles? Protein. Bone is made up of collagen protein. So if you just observe in our body, everything is like protein only. The nature of almost everything is like almost protein only. So therefore, my dear friend, is it possible to learn the modification of every protein? Is it possible to learn the modification of each and every protein? Not at all possible. So therefore, you have to learn for the exam the modification of collagen protein. Which protein? Collagen. And now please write down there, yes, the post-translational modification of collagen. Likho sir. What is the modification in collagen? Hydroxylation. And where is hydroxylation taking place? At lysine and is khatarnak English. Look here, my dear friends, what is collagen? It's a protein. Protein means what? Polymer of amino acid, made up of many amino acid. And somewhere in the middle you are having an amino acid known as lysine. Again it is made up of amino acid. Somewhere in the middle you are having proline, something like that. Understanding? So this is like our imaginary diagram, sir. So collagen is made up of like many protein, many amino acid. I repeat again, collagen is a protein, and we have already learned yesterday in detail what is a protein made up of. A polymer of amino acid. And somewhere in the middle we are having which amino acid? Lysine. And somewhere in the middle we are having proline. And on this, what is happening on lysine and proline, guys? Hydroxylation. And my students are expert. What is the meaning of hydroxylation, guys? Addition of OH group. So OH group is added here. OH group is added here. So, oh. Now read the statement. Hydroxylation at lysine and proline. You people know very well what is the name of the enzyme. Hydroxylase. Complete name. Lysyl. What is the name of the enzyme here also, sir? Hydroxylase. Complete name, proline. So which enzyme is helping in addition of OH group? Yes, it is hydroxylase enzyme. Lysyl hydroxylase, prolyl hydroxylase. And remember, these two enzymes will be requiring coenzyme, and that coenzyme is vitamin C. And that is vitamin C. Oh my God. Please turn back the pages and see yesterday's class. I taught you about vitamins topic. And in vitamin, I taught you the last one, vitamin C. देखो देखो टर्न करके देख, टर्न द पेजेस एंड सी विटामिन सी फर्स्ट फंक्शन हमने क्या लिखा था? The first function that we wrote there yesterday was, yes, it is helping in post-translational modification of collagen. Can you see vitamin C helping here? Yes. Now after making a chain like this, not only one, you have to make like 3 chains like this. And after making 3 chains like this, first do addition of OH, addition of OH, addition of OH, wherever lysine, proline add kar do. And now what you have to do, these 3 chains, you have to make them helical. 1 chain here, 2nd chain here, and the 3rd chain here. And that is known as the triple helix. And triple helix, yes, it is done by which enzyme? That is important, sir. And that will be your lysyl oxidase. And this enzyme recently asked in MC exam, it requires copper. This enzyme requires what, sir? Copper. Copper चाहिए उसके लिए. ठीक है जी. तो कोलाजन पहले एक चेन बनाओ, फिर उस चेन के ऊपर OH ग्रुप ऐड करो, and after adding the OH groups in that, make a triple helix of that. And triple helix is formed by lysyl oxidase. और उसके लिए क्या चाहिए, sir? Copper. Now your collagen is completely ready to be utilized in body. So in our body, the most common protein collagen, easily only you have to remember the modification of collagens. Are you done with that? Are you all done with that? So these are all like, you know, small topics that you need to remember, guys. One is about the codon, another one is about the RNA, and another one is about the modification of collagen. बच्चों come on, हो गया? Are you done? Please confirm me. चलो sir. If this is done here right now, okay, one single point here right now, sir. DNA to RNA, and mRNA to protein. And after the formation of this protein, remember these proteins will be folded, sir. Protein folding. Can anyone tell me the protein folding is done by which substance? Yes, the protein folding is done by, it's all like miscellaneous points. Protein folding is done by which one? Yes, substances which are known as try to pronounce that. I don't know why many students will be saying chaperon karte rehte. It's not chaperon actually, it is chaperons, chaperons. Yeah, it's not chaperon, it's chaperons. Okay, खतरनाक लोग होते हैं सर इंग्लिश में तो, ओ माय गॉड. ओके, रिसेंटली आई वाज एट वन ऑफ द एयरपोर्ट, मैं कहीं पर था, एंड आई वेंट टू दैट वो होता ना KFC खाने गया था. आई थिंक सम पीपल आर हंग्री राइट नाउ, और भी मज़ा आएगा. So I went for eating KFC and all those, and in that one fellow came there and he was like, I want Zinger box, and in Zinger box don't give me Pepsi Cola, give me Virgin Mojito. Had there too much Virgin Mojito. It's Mojito, it's not Mojito. It is written as J there, but you have to spell as H. And similarly this one, sir, chaperon kar de. Not chaperon, it's chaperons. Okay, so chaperons are helping in what, sir? Protein folding में help करते हैं sir. Okay, and now the last and final thing, sir, blotting techniques. सर किर्गिस्तान कब आओगे सर? आप बुला लो, मैं अभी आ जाऊंगा भाई. There will be a northern blotting technique, okay, and then there will be a southern blotting technique. So there will be a northern blotting, and then there will be a southern blotting and western blotting techniques. Okay, and you just have to remember this blotting technique is useful for what? What is the function of this one? Northern blotting technique, remember, it is useful for analysis of RNA. Southern blotting technique is useful for DNA, and western blotting technique is useful for proteins. So that is the only thing that you have to remember in this blotting technique, sir, nothing else. Okay, so which blotting technique is used for what? Northern blotting is used for RNA, southern blotting is for DNA, and western blotting is used for the proteins. But how to remember without confusion for long term? Remember, in North India people love to eat roti like me. In South India people love to eat dosa like you, and in western countries the people love to eat neither roti neither dosa, pizza. Is it okay? So therefore remember, northern blotting it's for RNA, southern blotting will be for DNA, and then western blotting technique is used for the proteins. That's all. So if you remember this much information, that will be done about about the molecular biology. Molecular biology हो गया इसका. Is it okay? Done? And after this, my dear friends, we need to learn about the topic of enzyme. So enzyme topic will take another 20-30 minutes. It will be done, sir. So shall we complete that, or shall we take a small break? Sir, small break. Break. अच्छा चलो ठीक है सर. We'll take a break exactly of 15 minutes. Okay, it's 8:30 right now. We'll start at 8:45. 8:30 right now, sir. We'll start from 8:45. ठीक है. In enzymes topic, guys, first of all, what is the definition of enzyme here right now? We all know that enzyme is a substance which is going to increase the velocity of reaction. Enzymes are those which are going to increase the velocity of the reaction, guys. So enzymes are going to increase the rate of the reaction. Okay, enzymes are going to increase the velocity of the reaction. So these kind of words will be used for, or the speed of the reaction. तो जो वेलोसिटी या फिर स्पीड और रेट ऑफ द रिएक्शन बढ़ाएगा, जो इंक्रीज करेगा, ठीक है. So that substance is known as the enzymes. So enzymes are those substances which are going to increase the velocity of the reaction. Done. Now the next thing that you need to learn after definition is nature of enzyme base. So please write in the heading there as nature of enzyme. Now what is the nature of enzyme? We all know enzymes will be protein in nature. What is an enzyme made up of? Protein. But along with the protein part, there will be sometime even the non-protein part also made up of this. And remember, yes. Then we need to learn okay, what is the name of the protein then, sir? Remember, the protein part of the enzyme is known as apoenzyme. The protein part of the enzyme is known as apoenzyme, and the non-protein part of the enzyme, it will be known as coenzyme. Okay, the protein part of the enzyme is known as apoenzyme, and non-protein part of the enzyme is known as coenzyme. And remember the another name of this one, the coenzyme is known as prosthetic group. The coenzyme, it is also known as the prosthetic group. Or else it is also known as the cofactor. So whatever coenzyme I'm telling you right from the beginning of the biochemistry, guys, what is the meaning of coenzyme? Coenzyme means it is the non-protein part of the enzymes. So normally proteins, enzymes are protein in nature. That protein part is known as apoenzyme, and the non-protein part is known as the coenzyme. And remember, these two together, apoenzyme and coenzyme, protein part and non-protein part together, it will be known as the holoenzyme. It is known as the complete enzyme or the whole enzyme or holoenzyme. So the protein part will be known as the apoenzyme and the non-protein part is known as the coenzyme base. And together it is known as holoenzyme. And that was asked in the MC exam. Take care about that. Okay? And that completes the definition, that completes the nature of enzyme. And now welcome to the most important thing, that is the factors affecting the enzymes. Number one factor that will be affecting the enzyme will be concentration of enzyme, and number two will be the concentration of substrate, number three will be concentration of product, and number four will be the temperature, and number five will be the pH. These are the five factors we need to learn which are affecting the velocity of the reaction. And now my dear friends, please help me right now. You have to help me right now, guys. For example, there is some reaction, substrate is getting converted to product in the presence of enzyme. In general, some reaction is going on. There is a substrate getting converted to product in the presence of enzyme. Now for example, what is the meaning of concentration, guys? Concentration means what? Amount. In simple English, concentration means what's the amount. If concentration of enzyme increases, अगर मान लो कि amount of enzyme बढ़ता है, if concentration of the enzyme increases, amount of enzyme increases, concentration of the enzyme or the amount of enzyme increases. Now tell me what will happen to velocity? Velocity बढ़ेगा कि कम होगा? Velocity of the reaction will be increasing or decreasing? Yes. From the definition itself, we'll know that what is an enzyme? Enzyme is a substance which is going to increase the velocity of the reaction. अगर आप enzyme बढ़ाओगे, if you increase more enzyme, then what will happen? More will be the velocity. So therefore, it is directly proportional to velocity of reaction. Or simply you can write as capital V. So more the concentration of enzyme, more will be the velocity of reaction. Similarly, if you increase the concentration of substrate, then what is going to happen? More the अगर मान लो if I'm having 10 substrate molecule, out of that 10, 8 are converting to product. If I increase that to 100 molecules, okay, and out of that 180 molecules are getting converted to product, okay, that means that more the substrate and more will be the velocity of the reaction, sir. So therefore directly proportional to velocity. And now welcome to the last and final condition right now, guys. What about the product right now? अगर आप product बढ़ाओगे तो क्या होगा? If you increase the product, then what is going to happen? Velocity of the reaction will decrease. Why? Why? Because it's common sense. When I'm already having like more product, will I think of making or increasing more velocity of the reaction? No. Already I'm having a lot of product there. So therefore, more the product, less will be the velocity, guys. Inversely proportional. So that means the final conclusion, more the enzyme, more will be the velocity. more the substrate, more will be the velocity, but more the product, less will be the velocity of the reaction, guys. That's all. So this you have to remember. And now what about temperature? अगर हम temperature बढ़ाएंगे तो velocity क्या होगा? बढ़ेगा कि कम होगा? If you increase temperature, what will happen to velocity of the reaction? For doing that, let us just take a graph here right now, guys. On the x-axis, I'm going to take temperature, and on the y-axis, I'm going to take the velocity of reaction. I mean to say capital V. Now, my dear friends, what is going to happen here is that when you are increasing temperature, that means what is going to happen, sir? You are giving heat energy, and whenever you are giving energy to anything, it will become more activated and it will be increasing. So therefore, when you increase temperature initially, yes, velocity increases. But if you further increase temperature, you already know, yes, you already know the nature of enzyme. It is actually protein in nature. अगर आप और temperature बढ़ाओगे, the protein will undergo denaturation. And when the protein is denatured, that means enzyme is gone. When the enzyme itself is gone, will the velocity increase? Ulta, it will decrease. So therefore, my dear friends, with respect to temperature, what is going to happen? You know, initially velocity increases, reaches to maximum. If you further increase temperature, velocity decreases. So never say increase or decrease. Yes, always say that you'll be getting a bell-shaped graph. With respect to temperature, don't say it is increasing or decreasing. Don't say that. Always say you're going to get bell-shaped graph. That means first increases, reaches maximum, then decreases. Okay. And my dear friends, you can clearly see here on y-axis we have taken what's the velocity, and this is the place where you are getting the maximum velocity. So this one will be known as the Vmax, maximum velocity. That is the place where you are getting the maximum velocity. Okay. And with respect to pH, exactly copy paste. Exactly आपको ना क्या करना पड़ता है? Copy paste करना पड़ता है. You need to do the copy paste. So therefore, my dear friends, on the x-axis take the pH, and on y-axis you can take the velocity of reaction. Capital V. Okay. And now what is going to happen with respect to pH? You already know the pH scale. pH scale will be like, you know, that 1 to 14, you know, acidic, basic, neutral and all. तो जैसे आप pH बढ़ाओगे, what is going to happen? Initially velocity increases, then reaches to the maximum, then again it is decreasing. Again you are getting bell-shaped graph, and here is the maximum velocity, sir. Vmax. So this topic is like so simple, guys, here right now. Yes. Concentration of enzyme increases, velocity increases. Concentration of substrate increases, velocity increases. Concentration of product increases, velocity decreases. And with respect to temperature and pH, we are getting bell-shaped graph. First increases, maximum, then decreases. That is what you have to remember. Okay, done. And now let us work out in detail with the concentration of substrate here. Ye wala part. This is something important. Okay, let's work in detail. Please write down the heading there as Michaelis Menten graph. Michaelis Menten graph. Michaelis Menten graph will give you the relation between concentration of substrate and the velocity. And you already know concentration of substrate is directly proportional to velocity of the reaction. So if it is directly proportional to velocity, let us take a graph. On x-axis I am taking substrate concentration. On y-axis I am taking velocity of reaction. Is it okay? And we can clearly see there, yes, what is the relation between substrate and velocity, guys? Directly proportional. So therefore what is going to happen? As you increase substrate, velocity will also increase proportionately. Therefore you will be getting a parabolic graph like this, guys. Okay, it is increasing proportionately. And in this graph, where are you going to get the Vmax? Yes, somewhere here. This will be the maximum velocity. Wow, very simple, nothing to worry. Unnecessarily students will be getting feared with, you know, graphs and all. Nothing to worry about the graph, guys. Michaelis Menten graph is giving you the relation between substrate and velocity. And remember, you will be getting a parabolic graph, directly proportional. And here, the most important thing here will be the constant. There is a constant which is known as the Michaelis Menten constant. Michaelis Menten constant. It is represented by Km. Michaelis-Menten constant is represented by Km. Now what is the definition for this Michaelis-Menten constant? Write down there. It is the concentration of substrate at half the maximum velocity. Michaelis-Menten constant Km is actually the concentration of the substrate at half the maximum velocity. Concentration of the substrate at half the maximum velocity. We'll do one thing here. Read that definition word by word in English and apply on the graph. Let's apply on the graph. What is the condition given? Half of the maximum velocity. Okay. Maximum velocity यहाँ है. Half of the maximum velocity somewhere in the middle here right now, sir. Okay, that is V max by 2. Half of the maximum. At that, it is the concentration of substrate only. Acha. So from here, welcome to the concentration of substrate. Okay. So this concentration of substrate itself is known as what? Km. Okay, that's all. It's so simple. Is it okay? So guys, make your life very simple here. What is Michaelis-Menten constant? It is nothing but substrate concentration only. It is nothing but concentration of substrate only. But when at half of the maximum velocity, that is an important condition. Is it okay? So आपकी नज़र ना, you people have to always have, in your mind these four things: concentration of substrate and Km on x-axis, whereas the velocity of the reaction and the Vmax will be on the y-axis. Clear? So Km is nothing but the concentration of the substrate at half the maximum velocity. Km is nothing but the concentration of substrate at half the maximum velocity. Okay, done. And now you might be thinking, sir, what is the use of this one? चलिए सर मैं आपको बताता हूँ इसका use क्या होता है. What is the use of this one? Please write down the heading there as next topic, enzyme inhibition. Enzyme inhibition. There will be like two types of enzyme inhibition. Number one, it will be like a reversible inhibition. Another one is irreversible inhibition. Enzyme inhibition ना दो तरह का होता है. One will be reversible enzyme inhibition. Another one is irreversible enzyme inhibition, guys. And I want all of you to please, okay. I want all of you to, yes, please stop writing here right now, guys, and please observe here right now. Reversible and irreversible, okay, enzyme inhibition. No need to even write down. It's there in the name itself. I'm having substrate. It's getting converted to product in the presence of enzyme. Normally reaction is going on. I mean to say normally means what, sir? Normally means what? Without inhibitor. Abhi inhibitor nahi hai. Now we are going to introduce the fourth player here, that is inhibitor. Inhibitor is coming here, inhibiting the enzyme. Enzyme inhibited and the reaction will be stopped. Okay, normal, okay, understood. Inhibitor will come there and inhibit the enzyme, therefore reaction stopped. But after some time that inhibitor will withdraw from there, so the inhibitor will go away from there. If it is going away from there, again the enzyme will start. Which type of inhibition? Reversible. Understanding here? So inhibitor coming, inhibiting, and again it is going away from there. So therefore, which type of inhibition is this one? Reversible inhibition. No need to study anything else. Now second category, we are giving a very strong inhibitor, and it is such a strong inhibitor, it is going to inhibit this enzyme forever. It's not going away from here. That inhibitor is not withdrawing from there. So therefore that is irreversible inhibition. It is inhibited forever. So therefore, my dear friends, enzyme inhibition दो तरह का होता है. It will be of two types: reversible, irreversible. You don't even have to worry about their definition. They will never ask you in the exam. It's there in the name itself. What they will ask you in the exam, and they will ask you pakka in the enzyme, yes, is welcome to reversibles. Reversible will be the competitive and non-competitive inhibition. So again, it is divided into two categories. Okay, one will be the competitive inhibition, another one will be the non-competitive inhibition. Okay, sir. Now what is the meaning of this competitive and non-competitive inhibition? Yes, that inhibitor is actually competing with the substrate. Inhibitor is competing with the substrate. Now what you have to remember, just two golden words that you have to remember for competitive and non-competitive. Yes, remember, in competitive inhibition, my dear friends, Vmax will remain same. The Vmax value will not change. What do you mean by remaining same? Before adding inhibitor and after adding inhibitor. So before adding inhibitor, we are calculating Vmax value. After adding inhibitor, we are calculating Vmax value, and it is the same. But whereas in competitive inhibition, what will happen? Vmax will remain same, Km value will increase. These two golden words you have to remember for competitive. And for non-competitive inhibition, remember exactly opposite. Km value will remain same before inhibitor, after inhibitor, whereas the Vmax value will decrease. Exactly opposite of that. So remember one thing, opposite will automatically come, sir. And I'm guaranteeing you in the June exam, coming June exam, pakka questions, definite question. You have to remember these two words, sir. In competitive inhibition, Vmax same, Km increases. And then in the non-competitive inhibition, yes, what will happen, sir? The Km remains the same and the Vmax decreases. Are you okay with that here? Examples, someone is asking me examples. Guys, examples you will study in pharmacology. Is it okay? In pharmacology, you will be giving drugs. For example, you are giving allopurinol. Allopurinol is a drug. So that drug, allopurinol, it's acting like your inhibitor. Allopurinol is a drug. It's acting like your inhibitor. And that is going to come and inhibit the enzyme. Which enzyme? Xanthine oxidase. And that is used for the treatment of what? That is used for the treatment of gout. Understanding here? So there they will tell you that allopurinol is acting like a competitive inhibitor, non-competitive inhibitor and all those things. Okay na? So now we cannot go in that examples of pharmacology. Otherwise we have to tell you about the drug, then it is used for which disease. That's totally a different story in pharmacology. So here in my biochemistry, learn the concept and apply the concept in your pharmacology next subject. Understanding, my dear friend? In biochemistry, pakka definite question in the June exam. Take my words. Competitive inhibition, they will ask you Vmax same, Km increases. Non-competitive inhibition, remember Km same, Vmax decrease. Vmax value will decrease. Is it okay? Come on. Are you done? Very good. Now welcome to the last and final topic in enzymes, guys. Please write in the heading as isoenzymes. Guys, what is the meaning of iso? Iso means same, not single. Iso means same. Okay, it will be the same enzyme. Very good. It will be the same enzyme, but in different tissues. I'll give you example. Listen here right now. Example. The best example for the isoenzyme is LDH. You know LDH? Lactate dehydrogenase. And lactate dehydrogenase has got 5 isoenzyme. LDH1, LDH2, LDH3, and then LDH4, and then LDH5. It is the same enzyme only, LDH only. It will do the same reaction, pyruvate to lactate, lactate to pyruvate. हमने already पढ़ा है. So it will be the same enzyme and it will be doing the same reaction, but it will be differing in the tissues. Which tissues? Remember, out of all this category, LDH1 and LDH2 will be more in your heart, whereas LDH4 and LDH5, this will be more in the skeletal muscle. Okay? And remember, LDH3, this will be more in your, present in brain, RBCs and all those things, kidney and all. Understanding here? Various organs. So what I am trying to tell you is that it will be the same enzyme doing same reaction. Okay, na? Same enzyme doing same reaction, but in different tissues, sir. So LDH1 and 2 will be in the heart, and LDH4 and 5 in the skeletal muscles and all. No, Divya, you are giving a wrong example. I told you hexokinase, glucokinase are two separate enzymes. They're not like isoenzymes. No, not at all. Don't take it wrongly. So this is an example of, like isoenzymes and all. Now listen here right now, sir. 1 second. Now why this topic is important, I'll tell you here. It is important because of clinical conditions, sir. Okay, now LDH is important, the lactate dehydrogenase. We have learned this in the carbohydrate metabolism, anaerobic glycolysis. Now this LDH enzyme is important. Come on, if you have learned pathology, myocardial infarction. Guys, in normal people like you and me, LDH1 will be more when compared to LDH2. In normal people like you and me, LDH1 will be more when compared to LDH2, sir. If you do a test right now in your body and my body, this will be done. Suppose a person got a heart attack, myocardial infarction. Yes, what will happen? Suddenly it will be flipping. In MI, in MI what is going to happen, guys? LDH2 will be more, guys. LDH2 ज़्यादा होता है, ठीक है? So in MI what is going to happen? LDH2 will be more. Are you getting my point here? And that is the reason why the moment someone is getting heart attack, you just do a blood test and you will see, अच्छा LDH2 ज़्यादा हो गया, declare, अच्छा इस patient को ना अभी recently heart attack हुआ है, something like that. So that will be useful for you in your pathology, study that. But in biochemistry, what is my concept here right now, sir? You have to remember how many isoenzymes? 5 isoenzymes. Heart will have LDH1 and 2, and the skeletal muscles will be having LDH4 and 5. Perfect, done. Now last and final, one more example important is creatinine kinase, CK, and this will be having like 3 isoenzymes: creatinine kinase MM, and creatinine kinase BB, and the creatinine kinase MB. What is the meaning of that, sir? MM means it is found in the muscle, skeletal muscle, and BB, this will be found in the brain, and yes, again this MB, sir, creatinine kinase MB, will be found in the heart. And फिर से वही, go into your second year, learn pathology. In pathology, myocardial infarction, जब heart attack होता है, तो this enzyme will be elevated in the blood. So all this will come in your clinical condition. So these two examples of isoenzymes are important. LDH has got the 5 isoenzymes, and then CK, that is creatinine kinase. Creatinine kinase has got only 3 isoenzymes: CKMM, CKBB, and CKMB, sir, in the skeletal muscle, in the brain, and the heart. Are you done with this one here? So these are all the topics that you need to learn about the enzyme topic completed, sir. Okay, so isoenzyme, and then enzyme inhibition, reversible, irreversible, competitive, non-competitive, and then you have to learn about the Michaelis-Menten graph and constant, and then you have to learn about all the factors affecting the enzyme activity, nature of the enzyme, proteins, that is apoenzyme and coenzyme, and then lastly definition. So this is done with the topics. It's a very easy topic. And guys, I have sent you the LMR. In the LMR, last minute revision, I have given you the examples of non-protein. Okay, you remember coenzymes? Already many coenzymes we have covered. Vitamin C is a coenzyme for lysyl hydroxylase and all, है ना? Biotin is a coenzyme for 3 enzymes. You remember? Vitamin B12 is a coenzyme. Remember? So my dear friends, that is nothing to teach over there. You have to remember which enzyme requires which coenzyme. For example, pyruvate carboxylase, it requires biotin. So you need to memorize that. So in that LMR last minute revision, at last I have given you a list of enzyme and the cofactor, enzyme and the coenzyme, enzyme, coenzyme. There is a table made, sir. And not only that, in that LMR, if you see, just in 3 pages, entire biochemistry covered, sir. Okay, now we are done with the rate limiting enzyme. Okay, yeah, of course, copper is a covalent. So we are done with the rate limiting enzyme. In one table, I have given all rate limiting enzyme. In one table, I have given all the locations. In one table, I have given you all the important amount. So do one thing, take a print of that and please stick that wherever you are studying. आप जहाँ पर भी पढ़ाई कर रहे हो ना, वहाँ पर आपके ना स्टिक कर लो वहाँ पर, ठीक है ना, and just keep on revising that and you will never forget that one, sir. Okay, so please take a print out of that one. Only 3 pages, sir. Only 3, not even like 3 pages, only 3 sides. Okay, just 3 sides. You just stick wherever you are learning and keep on revising every day, sir. List of all the location, list of all the rate limiting enzyme, list of all the diseases, list of all the amino acid and important products and all those things. So I think that will be like easier for you. Okay guys, and so please guys, okay, whatever I taught you, please learn that. That will be more than enough. I'm telling you, I have seen like thousands and thousands of students in my life. Again the same thing, guys. They will listen they will just listen to their friends and all. वो आकर बोलेगा अरे यार ये book में दिया है, that book is given that, this is book is given that, and again the same time after the exams it will be like, sir, all the questions came from your notes. You have seen like many feedbacks on my Facebook. So please, every time the same thing, sir. There will be hundreds of messages coming to my WhatsApp. Sir, इस book में ये लिखा है, इस book में ये लिखा है. Please guys, whatever I told you, please learn that. That will be more than enough. Is it okay? So please don't get diverted and don't get distracted and don't think negatively कि यार something is missing. So please don't do that. So therefore whatever I taught you, please learn it. Hope this session was useful to you people guys. Anatomy and biochemistry part is done, and we are going to stop the class here. Each and every topic that is required has been completed, sir. Thank you, sir. And anytime if you have any doubts, sir, please do one thing, drop a message in the that WhatsApp group में ना message drop कर दो, and that will be more than enough, sir. Okay, so whatever I've given you the notes, and please don't message me. One thing, please don't message me the doubts individually, guys. I'm sorry if I'm not able to answer you because there are so many messages in the WhatsApp. I can't answer them. Just you drop your message in the group. I'll surely come back to you. Okay. So please drop your message in the group only. Okay. Thank you. Okay. Thanks a lot, sir. I hope you people have enjoyed. Happy helping you all. I'm really happy that I've helped like so many students in different places. So please make use of these classes and try to revise everything, guys. Okay, and please don't misuse, guys. Okay, now please don't misuse. If you have recorded or something like that, please, I'm requesting you all, don't do that. It's very bad.